Hi, my name is Catherine McLaughlin and I am a Senior Application Scientist at Sepax Technologies. During this mini-webinar, I will discuss membrane protein separations using size exclusion chromatography. So on our agenda for today, we'll be going through some general information on membrane proteins and their importance in drug development. We will also go over the different size exclusion phases that Sepax has to offer: SRT, SRT C, and Nanofilm. From there, I will show some separations of standard proteins in different detergents, and then I will get into some real-world membrane protein detergent complexes and their separations to show the importance of having different stationary phases to choose between for these different samples. So what are membrane proteins? Membrane proteins are large biomolecules found inside the surface of cells. These cells are covered by a hydrophobic lipid bilayer, which stops things from going in and out of them. Membrane proteins sit in the lipid bilayer and help the cells communicate with their outside environment. The two types of membrane proteins are integral and peripheral. Integral membrane proteins are permanently attached to the membrane and can only be separated from the biological membranes using detergents, nonpolar solvents, and sometimes denaturing agents. Peripheral membrane proteins are attached to the lipid bilayer or to integral proteins by hydrophobic, electrostatic, and other non-covalent interactions. Later on in the webinar, I will show some examples of integral membrane proteins being analyzed by SEC. Research into membrane proteins is extremely important for a variety of reasons. For one, they are the target of about 50% of all small molecule drugs on the market. So their molecular structure is important for us to understand so that we are better able to design drugs that will bind to them. Also, there are several diseases that are caused by mutations in membrane proteins. These include depression, schizophrenia, and cystic fibrosis. Because detergents are often needed to extract integral membrane proteins from the biological membrane, any chromatographic step after extraction needs detergent present in order to keep the protein in solution. The type of detergent that should be chosen depends on the specific membrane protein, as well as the types of analyses that are going to be done downstream. There are 4 different categories of detergents. All of the detergents that I will show in the following examples are mild non-ionic detergents. Other types include bile salts, amphoteric detergents, and also ionic detergents, which are denaturing to proteins. In complex with the detergent, the membrane protein behaves larger than the hydrodynamic volume of the protein alone. This large size, along with the hydrophobic nature of these complexes, can sometimes add difficulties when trying to analyze them by size exclusion chromatography. Sepax offers 3 different size exclusion phases for the analysis of membrane proteins. These are SRT, SRT C, and Nanofilm. SRT is a 5-micron silica-based resin with a stand-up monolayer. SRT C is also a 5-micron silica-based resin, but with a lay-down monolayer which reduces access to the silica backbone. And Nanofilm is a 5-micron resin with a hydrophilic nanometer-thin film on the surface. Sepax's size exclusion phases are offered in a variety of pore sizes, and this ranges from 80 angstrom to 2,000 angstrom. Smaller 3 and sub-2 micron particle sizes, as well as a larger 10 micron particle size, are also available for scalability of our columns. All of these individual SEC offerings enable the selection of a better SEC surface chemistry for individual protein separations. The pore sizes we're going to focus on in this presentation are 300 and 500 angstrom. In the chromatogram you see on this slide, this is our quality control run that we do on any column before it's released. Without any detergent present, a standard protein such as MsbA, which is peak 4 in these chromatograms, elutes on the 300 and 500 angstrom columns in 8 and 10 minutes respectively. MsbA in complex with a detergent such as LDAO has a much larger hydrodynamic volume, which is why the elution time is much earlier, about 8 minutes on the SRT C SEC 500 as compared to 10 minutes on the same column which we saw on the previous slide, just without any detergent present. Size exclusion pore sizes sometimes need to be increased to consider the protein detergent complex. For this separation, the larger 500 angstrom pore size was chosen. And here we see that both SRT C and SRT give similar well-resolved peak profiles. We also ran two other commonly used protein standards. These are 158 kilodalton Aldolase and 440 kilodalton Ferritin. For the separation of Aldolase and complex with DDM detergent on a 300 angstrom pore size column, Sepax's SRT and SRT C both showed superior resolution over the nanofilm column. When we attempted to run the same sample on the larger 500 angstrom pore size column with another multi-side detergent, UDM, we lost the resolution that we saw previously of the aggregate and determined that the 300 angstrom pore size that we ran before this was more appropriate here. Ferritin in complex with DDM is too big for a 300 angstrom pore size. In this chromatogram, you see that the complex is eluting very near the void volume, around 5.5 minutes. It would be better to increase the pore size so that you could better separate out the aggregates and monomer. So when we ran this on a larger pore size, the 500 angstrom, Ferritin and its aggregates in the presence of UDM detergent were best separated by the SRT SEC 500 angstrom pore size. The separation between Ferritin and Aldolase gives a good visual of how different molecular weight proteins will elute with the same detergent and complex. Here we see that the SRT C SEC 300 separate both Aldolase and Ferritin in the presence of DDM. On Sepax's SRT C SEC 500, we achieved even better results. Both Ferritin and Aldolase are still well resolved, but the Ferritin aggregates are better separated as well. In the next few slides, we'll now go over some examples of some real-world membrane proteins in complex with a variety of different non-ionic detergents. Aquaporin water channels, Aqpz, transport water molecules across the membrane. Here is Aqpz in complex with octyl glucoside. This is a non-ionic dialysable detergent that is used for the solubilization and isolation of membrane proteins. This 23 kilodalton protein that should elute around 8 minutes is behaving more like 100 kilodalton protein. For this analysis, SRT C SEC 300 gave the best separation of these higher molecular weight proteins and also showed less secondary interactions. Sepax's SRT C SEC 300 gave the best recovery of the bacterial potassium channel that we ran here with DDM detergent. This 16 kilodalton homotetramer in the presence of DDM eluted off the column around 6.5 minutes, the same retention time as 670 kilodalton thyroglobulin would elute off this column without detergents present. While both the SRT and SRTC showed improved resolution of this Bacterial ABC transporter, MsbA, in detergent UDM, a larger 500 angstrom pore size may help with the separation of aggregates and monomers. Sepax's SRT SEC 500 showed better separation for the same sample just by moving up pore size. This is a 92 kilodalton purple bacteria photosynthetic reaction center. It's a trimer. For this membrane protein LDAO complex, Sepax's nanofilm SEC 500 provided the best separation as well as the best recovery. So in conclusion, Sepax's 3 SEC offerings for membrane protein separations, SRT, SRTC, and nanofilm, offer the opportunity to select a surface chemistry for the best possible separation and recovery of different protein detergent complexes. Sepax offers a membrane protein screening kit, which includes all 3 of these phase chemistries, so that way it's easier to find the best column for the membrane protein that you're working on. And the examples that we went over, the 16 kilodalton bacterial potassium channel and DDM had the best recovery on SRT 300, while the 23 kilodalton Aqpz and octyl glucoside had the best recovery and the best separation on SRT C 300. Then finally, for the larger 92 kilodalton Photosynthetic reaction center and LDAO, the best performance was found on the larger pore size Nanofilm SEC 500. Due to the large hydrodynamic sizes of the complexes, a larger pore size SEC column may need to be evaluated in order to achieve the best separation between the high molecular weight aggregate and the monomer. We showed the example of 65 kilodalton ABC transporter monomer, which exists as a dimer at 130 kilodalton. This should elute well on a 300 angstrom SEC column. However, due to the larger size of the protein detergent complex, a 500 angstrom pore size was necessary to achieve its best resolution. Sepax's SEC columns can be used for detergent screening and analytical high-resolution separation for specific membrane proteins. These SEC columns can scale up to larger particle sizes, such as 10 micron, as well as larger column sizes at 10 millimeter and 21.2 millimeter inner diameter columns for these membrane protein purifications that are done in protein crystallography processes. That concludes the webinar today. Thank you all for listening. If you have any questions that I did not hit on during this webinar, please reach out to our technical support team. The number is listed in the red box below, as well as an email address. And if you have any questions about pricing or sales, there's also contact information for our sales department. Thank you.